Hello, I'm Dr. Leanne Raymer, and today we're going to talk about gross anatomy and organization of the cerebral cortex. So the cortex is divided up in all kinds of ways, functionally, anatomically, histologically, and we'll talk about some of those. We're going to start with a general principle about the cortex, namely its gyrification. So the cerebral cortex is the dense collection of gray matter, the neurons and dendrites and their synapses that overlies the cerebral hemispheres. So it's important not to interchange cerebral cortex and cerebral hemisphere because anatomically they're very different things. Now, the cortex, you can see, is this layer of gray matter that lies on the surface of the cerebral hemisphere. So here's the whole right hemisphere, tracing with my pen, and you can see that it includes not only the cerebral cortex, but the white matter beneath it, and the deep brain nuclei, and even the ventricular spaces. So cerebral hemisphere, inclusive term, cerebral cortex, very specific term. Now the cortices of other mammals, like rabbits, lack sulci and gyri. And so the way we describe these smooth brains is lissencephalic, literally smooth-brained. Now the reason they have smooth brains is they have less area in their cerebral cortex for that dense collection of gray matter to exist. And I wonder sometimes if the phenomenon of lissencephaly doesn't underlie some human phenomena, not unlike the presidential debate of 2024. This brain is actually that of the domestic rabbit. So here we're seeing it in dorsal view in A and in lateral view in B. And you can see that in both cases, most of the parts of the brain that we recognize as a brain are present. So it looks like a brain that we recognize, except that the hemispheres are smooth. So they lack sulci and gyri. This is true in some congenital human diseases as well. Babies can be born lissencephalic. And I will point out the domestic rabbit, one of the sort of notable features of things like rabbits and mice and rats is their large olfactory bulbs. So they have really large olfactory bulbs relative to humans. Now in humans, of course, the cortex is highly convoluted, and named sulci and gyri cover the surfaces of the cerebral hemispheres. And we know that the sulcus, singular, or sulci, plural, are the little troughs or grooves, and the gyri are the little bumps or protrusions. Now, one of the purposes of the sulci, anatomically, is that they divide the hemisphere into lobes. And so the two main ones are shown here in dotted lines. The central sulcus, or the eponym is the sulcus of Rolando, or the Sylvian fissure, or the lateral fissure. So those are the two main and most apparent named sulci. Now, gyrification in the human brain increases through our pre- and postnatal development, and importantly, doesn't always produce the same finished product. So the actual width and depth and length of gyri and sulci are variable across individuals. Here you can see at 25 weeks of fetal development, there's a lateral sulcus and a central sulcus. So those are present and persist. And those are also conserved in most mammals. When we get into more elaborate gyrification patterns, that's conserved across most primates. And then we get into later periods of gestation as gyrification becomes more and more elaborate. It's species-specific differences. And then after birth, individual variation across people's brains begins to emerge. And so to me, this is just the culmination of genetics and experience in interplay determining what brain we actually end up with. But it's important to realize that although we have these named sulci and gyri, they certainly don't look anatomically identical across people. If you take two brains, you will find that the precentral gyrus, the primary motor cortex, is probably notably different in shape and length and width. So we measure the extent of folding in a bit of cortex using the gyrification index, or GI. And the gyrification index is just a ratio of the inner contour over the outer contour looking at the cortex. And those sort of lines are shown here in a section. So the outer contour is this dotted line, which skims over the surface of the hemisphere but doesn't dip into any sulci. And the inner contour, in contrast, is shown in black, diving deeply into every sulcus. And so if you place the ratio of inner contour over outer contour, of course you get the gyrification index. The pale line here is the white matter, gray matter boundary. So that's what that line corresponds to. So just pose a little question here. If you're thinking about this inner contour, which meningeal layer does that correspond to? If you think about your cranial meninges, which anatomical structure represents the inner contour? Little thought question. We can look at it, this is in 3 dimensions. This is what the inner surface of the brain, the inner contour revealed. This is the outer surface, or outer contour, outer shell. And by putting those in ratio, you can get a map like this, where higher GI corresponds to higher gyrification, or more cortical folding. So this is a little question slide for you. If you wanted to measure the gyrification ratio here, what 2 colors would you use in ratio? And as a side question, if you look at A, how would you measure cortical thickness in A? And cortical thickness is another measurement of the brain. So 2 things we tend to track quantitatively in imaging studies for different reasons: the gyrification index in different regions of the brain, and the cortical thickness in different regions of the brain. Now, when you track these things, not perhaps too surprisingly, the gyrification index decreases throughout life in healthy aging. So HC here stands for healthy controls. That's who we're looking at in this study. The gyrification index is plotted against age in years. And so you can see when we're very young, we have a highly folded, highly convoluted brain, but also we lose GI. That decreases very quickly. In older adulthood, and my definition of older adulthood is rapidly adjusting day by day here, let's take 40 to 50 as adulthood, and you can see that yes, our GI ratio is lower. However, our GI index, so is our rate of decline in it. So we've got less folding, but we're also not smoothing as quickly. Now, this has been used to study differences across brain in all kinds of populations. Here's a study cited. In this study, they're looking at psychiatric illnesses. So major depressive disorder, bipolar disorder 1, and schizophrenia. And you can see just from looking at the overlay with healthy controls that bipolar disorder 1 and schizophrenia are associated with a reduced gyrification index. Plotted here on this bar graph and with the statistics for you, you can see they're about the same under age 18, and then a divergence emerges. So there's a change in these 2 populations particularly, and they have a lower GI compared to both people with major depressive disorder and healthy controls above 18, and again, maybe even more pronounced, above the age of 40. Now, we don't know what this means, right? We don't know if it's a cause or an effect of the disease. The authors of this study point out that a lot of these illnesses emerge sort of around or after late adolescence. And so it's an interesting finding. It's one of the things we track using imaging studies and it can be used perhaps to predict the onset of a psychiatric illness or dementia, or to facilitate diagnosis. So next we're going to talk about how we divvy the cortex up. And there's functional and structural and anatomical divisions. And not surprisingly, because structure is related to function, all of these are interrelated. So I've said the first division is these 4 major lobes. And we use sulci to divide them. Again, here's the central sulcus of Orlando. If you follow the central sulcus, it is typically the longest continuous sulcus that passes from the central or the sagittal sulcus that divides the hemispheres all the way to the lateral sulcus. So it's typically the longest one traveling in that direction down the lateral surface of the brain. And again, those 2 crucially divide the lobes. Doobie-doobie-doobie-doo. And there's another landmark. This is the preoccipital notch, this little divot at the anterior aspect of the occipital lobe. And it's another dividing point. And it works in combination with the landmark you can barely see here. And that landmark, actually I'll skip to this slide for just a second. Maybe I'll just skip right along, is the parieto-occipital sulcus. So it's right here. So it's very visible on the medial surface. And then you have to extend it over on the lateral surface to finish divvying up the brain. So here in this specimen, you can see how visible the parieto-occipital sulcus is. And it forms the division between the parietal lobe and the occipital lobe. Now, here's the parieto-occipital sulcus as you extend it over the lateral surface. Doot, doot, doot. And then you have to draw some imaginary lines between sulci. So the first line you draw is the parieto-occipital line. And it goes from the parieto-occipital sulcus to the occipital notch. And so that parieto-occipital line, and it's sometimes called the lateral parieto-occipital line, but I don't like that term because it's confusing. The parieto-occipital line is an imaginary line between those 2 points, and it separates it creates the anterior border of the occipital lobe. The second imaginary line is here, this blue dashed line, boop, 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 and it's the occipito-temporal line, and it's an imaginary line connecting the back of the sylvian fissure to the parieto-occipital line. So you've got 2 parieto-occipital line, occipito-temporal line, and you just remember it in what it connects: parietal to occipital, occipital to temporal. And those are the imaginary lines that complete the division of the lobes on the lateral surface of the cerebral hemisphere. So let's come back to this slide for a moment. This is the limbic lobe. Lobe is in quotes there because it's really properly now called the limbic system, although people use both terms. And that's because it's a functionally related group of structures visible on the medial surface of the brain that aren't necessarily anatomically contiguous or continuous, but work together. This slide shows a number of the structures that most people agree are part of the limbic lobe. So the first is the cingulate gyrus. Now, I'll trace around the cingulate sulcus to show you the cingulate gyrus, this purple C. And the second is the parahippocampal gyrus on the inferomedial portion of the temporal lobe in green. And everybody agrees that those 2 are critical parts of the limbic system. Then people start to throw additional bits in. This classification shows the paraterminal gyrus, and the subcallosal gyrus here. And so those are often included along with other bits of brain like the amygdala as parts of the limbic system. And here's some easy warm-up questions. Hopefully they're walk in the park for you now. Just reminding you, refreshing you to think about every time you look at the brain. Which cerebral hemisphere am I looking at? Where's the front? Where's the back? Etc. So every time you look at a hemisphere, orienting yourself. Now, the major anatomical divisions of the brain and the CNS can be simplistically described as functional divisions. So this is the kind of diagram we all start with when we study the nervous system. And we might think of the frontal lobe as being the subset of the seat of executive function, right? Of planning and higher order thinking and our personality and such forth. The limbic system, which we've just spoken about, mediating things like memory and olfaction. The medulla, often described as the heart of cardiovascular and respiratory reflexes. So really simplistic, gross delineations of the brain. Couple of little things. This is a mid-sagittal section of the right side of the brain, and there is a little sulcus in the middle of the diencephalon. It's called the hypothalamic sulcus, and it splits the thalamus above from the hypothalamus below. And so if you're ever wondering where's the division between those two, there is a little tiny sulcus there in the wall of the third ventricle. This is a nice section because it shows this thin third ventricle and then the little cerebral aqueduct. If I can trace it nicely, opening into the beautiful triangle of the fourth ventricle. So it's a beautiful mid-sagittal section. And we talked about the parieto-occipital sulcus. There's all sorts of other named sulci and gyri on the surface of the brain. Now, another elusive bit of brain is the insula, or the insular cortex, which is sometimes called its own lobe. And you demonstrate the insula by prying open the lateral sulcus, which is what's happening here in A, and then cutting away a bit of the frontal, parietal, and temporal opercula. So the parts that cover the edges of the lateral sulcus. And so opercula literally means lid. So if you cut away that lid, you reveal the insula below. And the insula itself is still convoluted. It's got gyri in it, typically about two long gyri and 3 short gyri. But that's the insula inside the opercula. Now, within each of the major lobes, sulci and other landmarks create additional cortical divisions. And so here we've got the frontal lobe in this figure in A, and we're all familiar with the purple part, right? That's the primary motor cortex, contains the primary motor cortex. That is the precentral gyrus. So we're familiar with that. And we would say the precentral gyrus is bound posteriorly by the central sulcus and anteriorly by the precentral sulcus. So that's how we'd use sulci to delineate specific bits of brain. You can see other subdivisions here. There's a superior, middle, and inferior frontal gyri, in turn separated by superior and inferior frontal sulci. And even another set of divisions. So the inferior frontal gyrus has 3 named parts. The orbital part, which is near the eyes, so the most anterior part. The triangular part, which is usually taken as straddling this sulcus here. This is not a perfect depiction. And the opercular part. And the opercular literally mean covering the edge of the lateral sulcus is the opercular part. And so those are increasingly smaller subdivisions of the frontal lobe. Now, on the medial surface, there's another important subdivision. That's the anterior paracentral lobule. And the anterior paracentral lobule straddles the medial portion of the central sulcus, and it's the medial continuation of the primary motor and primary somatosensory cortices. So, a couple of thought questions for you. First, where is Broca's area in most people located here in A, and how would you describe its location anatomically? And second, what's represented in the anterior paracentral lobule, which is shown in B? So the anterior portion of the paracentral lobule. Now, in addition to the primary cortices, the cerebral cortex contains a lot of association cortex, which is responsible for higher processing. So I think I have a bigger image on. I'll go on to the next slide for a moment. So we tend to think about primary cortex when we think about functional brain areas, right? So primary motor cortex, for example, where motor commands originate from and exit the brain. Primary visual cortex, which is the first place in the brain that visual information arrives. Now, of course, if you just look at the schematic, you see right away that the primary cortices are by far the most minor territory in the cerebral cortex. So most of the brain consists of association cortex. You have unimodal association cortex, which is higher order processing largely for a single sensory or motor modality. So these secondary cortices, or unimodal association cortices, are usually adjacent to a primary motor or sensor area. And so if we take the primary motor cortex, we have a supplementary motor area adjacent to it. And we take the primary somatosensory cortex, try to make it a different pattern here, it's also purple. I'll stipple it. We have a somatosensory association cortex adjacent to it. It's mostly dealing with sensory information only, somatosensory information, which has arrived here, but then given a bit of context over here. So if someone detects touch in the somatosensory cortex, you might extend that to, do I like that touch? Was that enjoyable? Is it painful? A little more context, but still focusing on touch. Now, heteromodal association cortex is involved in integrating functions for multiple sensory or motor modalities. So it's even more integrative, and it tends to be surrounded by unimodal association cortex. So if we take our example of touch, once it has a little context around it, then it's going to be processed further in this heteromodal association context, our cortex, and integrated with things like visual information, emerging from the visual association cortex and impinging on it, auditory information, right? And so on. So these heteromodal areas are the most highly integrative. And you can tell, of course, a large portion of the frontal lobe, almost the entirety of the frontal lobe, is dedicated to integrative heteromodal association cortex. Now, in addition to its gross anatomical features, the organization and connections of neurons in the cortex has historically been used to create named divisions. And the feature of interest is the laminar organization, or columnar organization of neurons in the cortex. And long ago, people noted that the laminar organization varies across different parts of cortex. So here you see side by side a column of motor cortex and a column of sensory cortex, sort of a rectangle in depth dug out of the cortex. And you can see that the layers are sort of the same, with sort of the same functions preserved. Both have 6 layers. What's different is the relative thickness and density of neurons and dendrites in each layer. And Brodmann noted this, and sort of in the early 1900s, developed a scheme for classifying the brain based on cytoarchitecture. So these are Brodmann's cytoarchitectonic areas. They're still important because, not surprisingly, structure and function are linked. They turn out to be not a bad predictor of the function of the brain that we've studied in other ways through lesion studies and increasingly using imaging. And you can see here we've got 3 areas of cortex: the prefrontal association cortex in green, the primary motor cortex, of course, in pink, and the primary visual cortex in yellow. And these are Brodmann's areas 46, 4, and 17. And in columnar organization, although all 3 have 6 layers, they look visibly different. And that's what he based this structural map on. So it wasn't a functional map, but it happens to map fairly well to function. Now, when we divide the cortex and the brain into functional areas using either anatomical landmarks or cellular structure, we kind of imply a modular organization of the brain, where one portion of the brain is responsible for one function. And this idea is differently described as either localism or modularism or localizationism. You might hear all three terms used interchangeably. And this sort of model of brain function dictates that specific parts of the brain control a specific aspect of brain function. And so I'd like you to think about some historical or dogmatic examples of case or lesion studies that seem to illustrate localism, seem to indicate that this is indeed the case. Now, the opposite viewer model is more holistic or network-based, and you often hear this described as holism or connectionism. This is just the idea that specific aspects of function are governed by networks, in which different parts of the brain are all working together. So the historical seesaw has kind of tipped back and forth, alternating between first ascribing functions to specific areas in modularism, second, the relative importance of connections between areas as the seat of function. Part of that distinction has been based on the popularity and advent of new imaging modalities and new technologies to study the brain. So on the left here, we have a functional MRI with a bit of activity, a hot spot linked to a specific task. And on the right, we have a connectome, a model of all the connections in the brain and their relative weight and the relative size of each node in the network. And so these two areas sort of they fight for attention in terms of which interpretation is more important. Now, really, both of these models are useful, but the distinction between them is a little bit of a false dichotomy. So we all know that if you have a network and you knock out either one module or the connections between modules, function is altered or lost. So we now appreciate that complex neurological or psychiatric syndromes cannot be understood based on focal brain lesions. And this has been best illustrated in clinical studies where lesions in different locations produce the same clinical outcome. And so I'll show you an example of one of those. So this is a recent study in which they used the human brain connectome to localize symptoms from focal brain lesions. And this is the clinical problem right here, is lesions in different parts of the brain, seemingly totally disparate, one up here, one down in the temporal lobe, and one back here in the parietal lobe, causing the same symptom. And you can look at people with these discrete lesions and overlay them on either a map of anatomical connectivity, using something like DTI to show the connections between those regions, or functional activity, using something like resting state functional MRI to determine whether those regions are indeed functionally connected or functionally synergistically working together, and using that determine whether they are part of the same connected brain network. And this is a big field emerging using machine learning and AI to help predict and explain how lesions manifest clinically when they can be so variable. And it integrates our knowledge of anatomical and functional connectivity of the brain. Now, a third major feature of organization of the cortex is topography. And in fact, topographical organization is an important feature of the whole central nervous system. It applies to both cortex and subcortical structures, including white matter tracts and nuclei. Topographical organization, the definition is that adjacent areas on receptive surfaces or adjacent muscles are mapped to adjacent areas of gray and white matter. And you can have this for somatotopy, so different surfaces of the skin mapped to spatially related surfaces of brain. You can have retinotopy, audiotopy, and so on. So all of those are examples of topographical organization. Now, the best known topographic maps are the somatotopic maps of the sensory and motor cortices. And of course, these gave rise to the somatosensory homunculus in the postcentral gyrus, which is here shown in the left hemisphere of the brain in blue, and the motor homunculus in the precentral gyrus, shown here in the right hemisphere of the brain and in red. Now, Wilder-Penfield developed this map a long time ago, in 1930, using electrical stimulation of the cortex in awake subjects. And that's where we got this map from. In 2023, there was a major update to Penfield's somatotopic map for the motor cortex. And so the original map, here it is, proposed a continuous map of body to cortex that projected from medial to lateral. Of note, there was actually a large-scale replication of Penfield's work published in 2020, and that was the first time the question was really raised. And the question was, wait a second, when we replicated these experiments, unlike Penfield, we stimulated some regions of primary motor cortex that did not elicit movement or even the desire to move. And so there was something else in there, and that's been known for a few years. In 2003, this is the updated version of the map. It's produced from analysis of MRI data sets, both of humans and non-human primates. And what the authors found is the map has the same general organization. So if we break this up, the effector regions are still foot is medial, hand is middle, and mouth and face are still lateral. That's still true. But interspersed between these effector regions, or interleaved region, are intra-effector regions. And when these inter-effector regions, that's what's shown in blue. And these, when stimulated, did not produce, they're not direct mediators of movement. They're very different. What they all have in common is they project to the singular opercular network, and they're involved in things like movement initiation. And so movement planning, movement coordination, they are very different from the direct effector centers of Penfield's original model. So major update to the motor homunculus. And this is cool. This is one of those things that an updated technology, an updated approach to large scale analysis facilitated, a correction to a model that we've been using for more than 100 years in functional neuroanatomy.